Okay, so we started looking at the analogs, the base analogs, and we are talking about 5-bromouracil, and we said that this bromouracil is able to bond with adenine and also guanine through the process of tautomerization. So it's alternating between these two forms of keto form and the inner form. It's able to bond or form hydrogen bonds with adenine and also with guanine. So the mechanism of 5-bromouracil, as in the previous slide, could be translation 1, where we find it being an analog of thymine with a difference of bromine and the difference of bromine and the methyl that is found in the 5-bromouracil and thymine respectively of this formation here. So it's going to form bonds with the adenine. It's going to form the bonds with the adenine. And then we have also this example of the transition that happens between guanine and the 5-bromouracil, as explained in the earlier example. We also have the intercalating agents. We have the intercalating agents. So the intercalating agents, these are a group of aromatic organic molecules. A group of aromatic organic molecules. They have almost the same dimensions as the nitrogenous base pairs. And then all are actually flat. They have multiple ring molecules. So no wonder they are known as aromatic. They have multiple ring molecules, and these interact with the bases of the DNA. And so what happens as they are interacting between the bases of the DNA or with the bases of the DNA, they insert between them. So intercalating, they intercalate, they insert between the bases. And they are called intercalating agents. So they intercalate between the bases of DNA, and this is going to cause a disruption in the structure of the DNA. Now, this insertion causes a stretching of the DNA double strand, and the DNA polymerase, in a layman's language, will say that it becomes food in that it is going to insert an extra base opposite an intercalated molecule. Because the DNA polymerase will not know that, okay, there has been an insertion of this intercalating agent and there has been this stretching the DNA. And so it is going to insert an extra base that is opposite that intercalated molecule. So that causes an addition or a deletion of a base pair to that intact DNA. So there could be an addition or a deletion. So this is going to alter the reading frame of the gene. Remember when we looked at gene mutations, we talked about certain mutations that alter the reading frame, and we call these as the frame shift. So it means that these intercalating agents will cause frame shift mutations. They're going to cause frame shift mutations. So they can also block the process of replication and also the process of transcription. So the diagram there is just showing the intercalated molecule. And since due to the intercalated molecule, this can also result in production of a non-functional product. That could be a non-functional product, that is molecule that is produced. Okay. So these are the intercalating agents. Examples of intercalating agents include proflavin, It includes acridine orange. It includes ethylene bromide as well. Okay, just a minute, I bring back the screen. Has been a disruption. So we have so far looked at the physical agents. We looked at the physical agents or the physical mutagens. We looked at the physical mutagens. We looked at radiation, and we said radiation could be non-ionizing. As an example, we gave an example of UV light. We also looked at the ionizing radiation. And in this case, we looked at the X-rays and the gamma rays. We talked about their effects and how they cause mutations. And we're also looking at the chemical mutations. Also looking at the chemical, looked at the chemical mutagens. We've looked at the deaminating agents. We looked at the deaminating agents, and of importance, we looked at nitrous acid, the HNO2, which is a nitrous acid. We looked at its effect modifying an amino, it's converting an amino group to a keto group. And we've looked at the alkylating agents in that they add an alkyl group. And we've also looked at, after alkylating agents, we've looked at the intercalating agents. And also we looked at the base analogs. And we said that the base analogs will resemble a pyrimidine, the base analog will resemble a purine. And so it's going to insert where the original or the normal base was supposed to insert. This will lead to a mutation. So lead to a mutation. And then we have looked at the intercalating agents as well. The intercalating agents, which are going to intercalate or insert between the bases. Okay, so that's where we are. So just a minute so that I can project my screen. Okay, so examples of intercalating agents that we have ethidium bromide. Ethidium bromide. So this is used during agarose gel electrophoresis. So even during the process of PCR, if you remember, if you did the experiment or the lab on PCR, remember that after PCR you do electrophoresis and you add ethidium bromide during that process of electrophoresis. The other example that we have is a proflavin, and the other example that we have is acridine orange. So the intercalating agents. So remember, the mechanism said that they're going to insert between the bases. So in the example given, they have a template DNA molecule. You have a template DNA molecule, and proflavin is given as an example there. And we have intercalating proflavin molecules there in the DNA strand. As in this example, we have the template DNA proflavin, and this leads to a disruption in the DNA replication, a repair, or also a DNA recombination. So we have this intercalated proflavin that has inserted between the bases there. So this is how that proflavin causes the mutation, the mutant. And then we have chemical agents. Other chemical agents are bulky additional products. So these are huge molecules which are able to covalently bind to purines in DNA. This is going to lead to the deletion of such a purine from the DNA that is affected. So for example, aflatoxin B1 is a bulk addition mutagen, which is also carcinogenic, which is found in stored seed crops such as groundnuts and maize as well. And it is linked to cancer of the liver and also correlated to hepatitis B virus occurrence as well. So you have bulk additions. These are going to covalently bind to purines in the DNA. That will lead to the deletion of that purine in that DNA that is affected. So an example is aflatoxin, which is mutagen and also carcinogenic in nature as well. And then we have chemical mutagens, metal ions. So we have different metals such as arsenal, cadmium, we have chromium, we have nickel, and also their compounds, which are mutagenic as well. So these ions may be associated with the production of reactive oxygen species, species, which hinder the DNA repair pathway. And hence this is going to cause DNA hypermethylation and also can cause direct damage to the DNA itself. So these metals alter the replication mechanism of DNA. They alter the replication mechanism of DNA. So for example, we mentioned that they can cause hypermethylation. An example of such a metal that can cause hypermethylation is nickel. Then we talked about these metal ions affecting the repair processes. So an example or examples include cobalt, arsenic, nickel, and cadmium. These can affect the repair process of DNA, such as in the case of a mismatch repair and base and in a nucleotide excision repair. So we have chemical mutagens, which are metal ions causing mutations. The other thing is we have biological mutagens. We have biological mutagens. So examples of biological mutagens include viruses, bacteria, and transposons. So we are very familiar with the HIV virus, which is the virus which causes the disease AIDS. So we find that these viruses, for example, other viruses given rubella virus, we have cytomegalovirus, we have hepatitis B virus. The viruses insert their DNA into the human genome, and this is going to disrupt the insertion of that viral DNA into the human genome. Is going to disrupt the genetic function. So once that viral DNA has been inserted, the DNA then replicates and then transcribes and then translates the viral protein. So instead of our proteins being translated or being formed, what will be formed is the viral proteins. And so we find that these mature particles actually form in an exceeding cell. So some viruses actually cause cancer. So an example is a Rose sarcoma virus. It's going to cause cancer. So the viruses are mutagens in that they insert their DNA in the human genome, and then after it has been inserted, that viral DNA is going to replicate and then transcribe and then translate into viral proteins instead of our proteins. And then we have bacteria. For example, Helicobacter pylori. These bacteria cause inflammation, during which we have oxidative species which are produced. These are going to cause damage to the DNA, and this is going to also reduce the efficiency of the DNA repair system, and henceforth increase mutation as well. Are going to increase mutation as well. And then we have what are known as the transposons. The transposons, which are simply jumping genes. They are jumping genes. They are jumping genes. So they are non-coding DNA sequences. So they are going to jump from one place to a different place in an exceedingly large genome. And this is going to influence the function of the gene. Their insertion in the DNA, chromosomal DNA, is going to disrupt the functional elements of the genes. So the pictures are simply detecting what we've been talking about, for example, rotavirus, and causing an integration of the viral DNA in the human genome, and that leads to the replication, transcription, and translation of the viral genome. And bacteria are going to cause inflammation, which is going to cause damage to the DNA. So these are the mutagens that are there. These are the mutagens that cause non-spontaneous mutations. So mutagens. So do you